Hello, everyone, and welcome to the Parkinson's Foundation's PD Health at Home Wellness Wednesdays webinar. Thank you for joining us. We will begin soon. Please know we are recording today's presentation, and you will receive a follow-up email from us with a link to today's recording and additional resources in about 1 week. You can submit a question or a comment by using the Q&A feature. We will save questions for the end of our program today to offer to our presenter. We'll be together for about 1 hour, and thank you for joining us. PD Health at Home is presented by the Light of Day Foundation, whose generosity has made this programming possible. And welcome to all of you to PD Health at Home Wellness Wednesdays. Today we will learn about current studies that aim to improve, slow, or someday stop the progression of Parkinson's disease. Current Parkinson's treatment mainly focus on managing symptoms because there are no disease-modifying therapies available at this time. Research aimed at exploring therapies that could possibly change the course of Parkinson's is rapidly evolving. In this webinar, we will explore the possibility and the potential of disease-modifying therapies. Leading the presentation today is Associate Professor and Clinician Scientist, Dr. Lorraine Kalia. Dr. Lorraine Kalia is a movement disorder neurologist and scientist at the University of Toronto and Toronto Western Hospital. Her clinical and research work focuses on Parkinson's disease, where she leads a team studying the molecular drivers of neurodegeneration to help develop future therapies. She holds the Wolfond Crimbill Chair in Parkinson's disease research and serves as co-editor-in-chief of the Journal of Parkinson's Disease. Dr. Kalia, thank you for sharing your time and your expertise with us today.
Good afternoon. And I'm happy to be here and happy and thankful to be invited to share this hour with the group. I'm just going to share my slides and get them up here for everyone to see. And so thank you, Krista, for the introduction, and you've provided a wonderful introduction to what I'll be talking about. I will be presenting hopefully for just the next 45 minutes or so, and then we'll have some time for questions and answers from the audience. So please enter your questions so that we can have a conversation at the end. And as Krista mentioned, the focus for this afternoon is really looking at disease-modifying therapies, exploring the possibility of them for Parkinson's disease. And I'm talking to you from Toronto, Canada this afternoon. So what I'm hoping to get through in the next 45 minutes or so is first an overview, and Krista actually gave a really nice initial overview of understanding what are disease-modifying therapies. This is a term that many of us use, but I want to define that for all of us. And get a sense of what the current state is in the context of Parkinson's disease. Then I want to dive into telling you about really promising areas of research and how, in particular, there are many examples, but I'm going to focus on 3 specific examples, which are alpha-synuclein, GBA1 or glucose reversidase, and LRRK2, and we'll dive into that a little bit. And then lastly, I want to touch on, again, this will not be entirely comprehensive, but 2 key areas that I think are really important for further investigation and also as potential hurdles that we're facing right now in the area of research of disease-modifying therapies, and that's around getting drugs to the brain, so brain delivery and biomarkers. So we'll start off with our first objective, which is to really understand the current state of disease-modifying therapies, or DMTs, for Parkinson's disease. And I thought I would just take a step back. I'm sure many in the audience are very familiar with this term, but there may be some who aren't, and there may be some in the audience who think they know what it means, but maybe it's different than what most of us consider a DMT. And so I thought I'd just start by a general description or definition as to what a disease-modifying therapy is. Because in fact, these kinds of therapies aren't specific to Parkinson's disease. There are diseases, throughout medicine where DMTs are needed, in particular chronic diseases, such as Parkinson's disease, where you actually want to change the course of the disease. So here's one definition. It's provided by Dr. Cummings, who's actually an expert in Alzheimer's disease, that he provided many years ago in this publication. And it's a pharmacological treatment or other therapeutic intervention, because it doesn't necessarily have to be a drug, that impacts the underlying pathobiology of the disease. Sometimes people stop at that as the definition, but I think it's really important to add on the second part, the and, that you don't actually just affect the underlying biology of the disease, but it actually produces something measurable from a clinical trials point of view, but something meaningful for a person who has the disease that actually benefits the clinical course of the disease. And this is kind of just a generic term. You can imagine this being applied to conditions such as rheumatological conditions like arthritis. And so I put that out there just so we're all on the same page for this conversation. As you already heard, symptomatic therapies are really what we have in Parkinson's disease. And I just want to give a diagram as to what we mean by symptoms and what we mean by underlying pathobiology. This is a schematic, a way that I like to visualize Parkinson's disease, where along the bottom line, so along this x-axis of the graph, we have time. So 0 is when a person's diagnosed with Parkinson's disease. This is all the time after the diagnosis. This is all the time before. And then this this y-axis, I like to see as representing disability or impact on people's lives as a consequence of symptoms. And the symptoms I have categorized, as many people do, into motor symptoms, which are the movement problems that come with Parkinson's disease, and they're represented by this blue triangle. Non-motor symptoms, which, as many of you know, are a huge range of different kinds of symptoms that can range from sleep problems to constipation to thinking and memory problems. And then on top of that, I also add this last triangle, which are complications related to having Parkinson's disease and being on Parkinson's disease medications. And these include things like fluctuations, dyskinesia, for some people, hallucinations. And each of these triangles, as you can see, increases over the course of the disease and accumulates on top of each other, so that the actual disability associated with the disease, or the impact that it has on someone's life, increases over the course of the disease. And these are the symptoms that pile on top of each other to result in that. Like you heard, we have a lot of symptomatic therapies, and I'm sure, like many of you are experiencing, you're probably on many of these. And I think the silver lining in terms of having a diagnosis of Parkinson's disease is that we actually do have a lot of symptomatic therapies to offer. Many that are listed here, levodopa, which is our typical gold standard medications, but a variety of others that act at the level of dopamine in the brain that some of you may or may not have tried, and amantadine that we often use for treating dyskinesias, and then other additional drugs that sometimes we use to try and help symptoms but don't necessarily affect dopamine in the brain. These are the drug therapies that we have, and some of these pills might be familiar to you. There's also more advanced ways that aren't via pills, where people can actually also boost the dopamine in their brain. For some people, that's in Canada, we call it duodopa. In the States, it's called duopa, which is actually delivering levodopa through a tube via a gel formulation of the drug. More recently, there's this subcutaneous or liquid formulation of levodopa that can be given through a needle and a pump in a somewhat similar way to an insulin pump that diabetics have. And then really experimentally is this idea of actually using stem cells that get implanted to the brain, and they produce dopamine to be able to deliver dopamine directly within the brain in a delivery method that is by a biological stem cell. And so all of these are really symptomatic therapies. They affect those host of symptoms that I've shown here, not all of them, but some of them. And as I mentioned, can also lead to some of the complications related to Parkinson's disease. There are surgical therapies that some of you may be familiar with or have experienced. So neurostimulation, in particular deep brain stimulation, being the typical kind of surgery that affords this. And then older and newer techniques to actually make lesions or burn areas of the brain to affect the circuit that's involved in Parkinson's disease. And previously, that used to be done with a similar electrode to what's used in DBS. And more recently, there are many centers that are using focused ultrasound to do the same kind of lesioning within the brain. And then, of course, there are the non-surgical and non-pharmacological therapies that can definitely have benefit for people with symptoms. They range from everything from exercise to arts-related type therapies. And these are obviously important complementary types of therapies that can help people with Parkinson's. But ultimately, I showed you what kind of sits on top here in terms of symptoms. But what is actually driving those symptoms or causing those symptoms is what's happening underneath, what's happening at the level of the brain in terms of the pathobiology. Ultimately, brain cells in Parkinson's disease are dying faster than the rate of normal brain cell death. As adults, all of our brain cells are actually dying. Um, uh, but in Parkinson's disease, certain ones die at a faster rate. And it's a variety of different areas of abnormalities in cells that lead to the pathobiology and lead to this brain cell death. And it's in this area where if we can actually affect that those processes, that one could imagine you could actually prevent brain cell death or at the very least slow brain cell death. And that's where disease modification has to target to be able to accomplish that. So I showed you a kind of generic definition of disease-modifying therapies. This is specifically for Parkinson's disease. I've just retooled it a little bit to, again, say that it's a therapy. It could be a drug. It could be an other therapeutic intervention, but it impacts the underlying pathobiology of the neurodegeneration or the brain cell death. And it produces, again, a measurable and meaningful benefit on the clinical course of the neurodegenerative disease. So another way to drive this home, another way from an illustration point of view to explain what a disease modifying therapy is for me to lay it out against also what it's not. So if we look at this arrow, which I want you to think as representing the progression of Parkinson's disease over time, it's this size. Symptomatic therapies are like a Band-Aid. So the disease is still progressing underneath, but we're able to alleviate some of the symptoms, kind of cover them up a little bit with a Band-Aid. That's what symptomatic therapies represent. We often think about a cure, and I put that here as a contrast, where what a cure would be the disease starts, and then we intervene, and we can actually go back in time to where the person didn't have the disease. So that would be what a cure would be. You actually never had a trace of the disease. And similarly, even better would be the concept of prevention, where you were destined to have the disease, but we had a way to actually stop it from actually starting. And that's to contrast it with the disease-modifying therapy, which I've represented here as a thinner arrow to indicate that the disease is still there, it's still progressing, but just slower or with less severity. And so this is what we're talking about. So this is an empty slide as an indicator and really a placeholder for disease-modifying therapies for Parkinson's disease. We have no disease-modifying therapies for Parkinson's disease at present. Nothing that I can prescribe to people in my clinic or that your physicians can prescribe to you. That's not to say that there's not a lot of investigation going on, and that's what we're going to talk about for the remainder of this talk. I do want to point out that although we have nothing for disease-modifying therapies, as I told you, disease-modifying therapies are something that are sought after for most chronic conditions. And sometimes I like to look to other areas of medicine for inspiration. And I have to say multiple sclerosis is a neurological disease that is very much inspirational in that this slide represents all of the, and this is actually not an up-to-date slide, all of the disease-modifying therapies available for multiple sclerosis. And so I put this up here for us to remind ourselves that this is attainable, and also to kind of demonstrate that in cases of neurological disease, that it is possible. And so sometimes people will often say, well, it's too hard to find a disease-modifying therapy for brain diseases because the brain is so complicated. And I completely agree that brain is the most fascinating and complicated organ in our body. But I do think multiple sclerosis provides us some evidence that even at a complicated disease that affects a complicated organ like the brain, there is a possibility of finding not just one, but many, many disease-modifying therapies. So I put that there for inspiration. So moving on to point two, I want to now dive into talking about promising areas that are going to give us hope and hopefully some actual new therapies around disease modification. And I will upfront just disclaim that I'm not going to cover the whole spectrum of what's out there in terms of research. So there may be specific drugs that you heard about and you were really hoping that I would talk about them today, and perhaps I won't. But I just want this to be a bit of a sampling and a couple of areas that I'm particularly excited about or have an interest in or have been involved in some way in research related to this. So I'm happy to take additional questions of things that maybe you didn't see in this part of the conversation. And what I really want to focus on is not necessarily all of the drugs that are out there for for research in the disease modification area, but to give you general principles as to when people talk about these different proteins in our body, so alpha-synuclein, glucocerebrosidase, and LRRK2, or these genes, GBA1 or LRRK2, that you have some basis for some understanding, because these are right now, I'd say, the very hottest areas of investigation in Parkinson's disease for new therapies. Going back to me explaining that I will probably not cover all of the drugs that all of you are necessarily interested in, this is partly why, because there are lots of drugs that are being investigated. This is an amazing summary of clinical trials that are going on for Parkinson's disease, written in part by Kevin McFarthing, who's a person with Parkinson's disease. And he's been doing this yearly in the Journal of Parkinson's Disease with a variety of colleagues in research and in patient organizations to allow us to have a yearly snapshot as to what is going on in the field. And I'll just orient you to this figure. The intention here is to summarize all of the reagents or compounds that are in clinical trials. This bottom half or so, kind of divided out along this line here, this bottom half has symptomatic therapies. And then this top half has disease modifying therapies. So, going back to that slide that I had, these therapies are intended to improve symptoms, and we need new treatments to improve symptoms for sure. So great, there's lots of investigation into that. And then there's also this whole area of investigation into disease modifying therapies. The color coding is related to the different mechanisms or different ways that the drugs are intended to act. And then each of these slices of the pie represents different phases of clinical trials. So phase 1, phase 2, and phase 3. And with that, I thought I would just pause a moment to just review what that means for those who haven't heard of these terms, or who have and maybe aren't certain exactly what they mean, or those who needed some reminders. Because for us to be able to get a drug in the clinic, it unfortunately, and fortunately, because we want what we bring to the clinic to be robust and safe, but it takes a lot of time. And the steps for investigation are outlined here. A lot of time actually is spent in the laboratory to really understand the disease, to understand what aspects of the process of neurodegeneration can potentially be targeted, to find new areas of investigation. And so that is a lot done at a laboratory bench, using ways to try and mimic the disease in a laboratory. So not in people, in cells in a dish, sometimes in animal models, to try and, as best as we can, emulate the disease and then try and fix it or learn about it. When we think we have something that is potentially effective enough, we have enough evidence from the laboratory studies, and potentially safe enough, again, evidence from laboratory studies, then those drugs can go into a phase 1 trial. And a phase 1 trial usually is actually just testing, if it's a brand new drug that's never been in people before, is to test if the drug is actually safe in people who are relatively healthy. And so these phase 1 trials are usually first-in-person trials to just see if there's any unexpected side effects, to see if there's side effects that we expected, and to start to understand how the drug affects the body, and potentially how the body affects the drug. Does the body metabolize it really quickly? What kind of doses can people tolerate? Once something looks okay there, it might go on to a phase 2 trial. And this usually involves looking at safety of the drug, not just in healthy people, but actually in the people that you intend the drug to be for. So in this case, it would be in Parkinson's disease people with Parkinson's disease. Just because a drug is safe in a person who doesn't have Parkinson's disease doesn't mean it might not have problems in people with Parkinson's disease, especially since people with Parkinson's disease are also taking other drugs, and you need to know if this new drug affects the levodopa, for example. And so phase 2 trials are focused on that, and they also start to look at whether or not the drug might improve the course of the disease or have some disease modifying effect. If it looks like that's the case, then usually it goes on to a much larger trial, which is usually a very large trial of hundreds of patients, which is a phase 3 trial, where really the major aim of the trial is to say, does this new drug reduce the progression of Parkinson's disease? Does it actually modify the disease course? And these tend to be longer trials because Parkinson's disease, although it's a progressive disease, is also not a very, very fast progressive disease. And so you need time to actually see if a drug is having any impact on that progression. Usually when things look good at a phase 3 trial, then drugs typically will end up finding their way then to getting approved and being able to be prescribed. And then phase 4 trials are usually added on to look for more safety. Often we want to know how things work in the real world as opposed to just a very kind of controlled trial or a controlled environment. So that's the layout of the kind of pathway for a drug to get from the lab to the clinic. And just going back to this figure again, you can see that at least for the disease modifying therapies, there's still many of the drugs are in this phase 1 trial. And just because you're in a phase 1 trial doesn't mean you'll make it to a phase 2. Sometimes many drugs will actually fail in a phase 1 trial and never see progression to phase 2. But I think the good news is we can see that there's a healthy number of drugs in phase 2 trials for disease modifying therapies, and some that have even are currently being investigated in phase 3. So what I'm going to focus on, like I mentioned, are kind of 3 different types of targets for these drugs, and they're highlighted here. So GBA, or glucose ribosydase, LRRK2, and this is ASN, which is alpha-synuclein. And you can see green, yellow, and purple, they're all kind of decorating the top of this graph. So there are a couple of drugs for LRRK2 and a larger number for glucose ribosydase, and a fair number looking at alpha-synuclein. And hence why I thought these were good areas for us to focus on. So we'll start with alpha-synuclein. For those of you who aren't familiar with this with this protein, this is a protein that we all have in our bodies. It's actually a quite abundant protein, especially in brains, and so it has what we call a physiological role, meaning it does something that needs to be done in our brains. We think that it's probably involved in releasing brain chemicals, and the way that the brain is structured when it's in its physiological or regular day job is it can sit by itself in what we call a monomer, so just one alpha-synuclein protein, or some people think it might actually sit in tetramers, which is just a small group of proteins to do what it needs to do. For reasons that may be numerous and are still being elucidated, in Parkinson's disease, there is a switch where these physiological forms of alpha-synuclein turn into what we call pathological forms of alpha-synuclein, meaning that they're harmful. And so when that happens, alpha-synuclein starts to form different kind of structures that we call protofibrils or oligomers. Some people call them ribbons. They can form what look like fibers or fibrils. And then they can take on a very kind of large aggregated form. This represents here a brain cell. And this yellow blob here is what we call a Lewy body, or this little squiggly line here is what we call a Lewy neurite. And that's basically a large aggregation of many, many proteins in different parts of the cell, but a good chunk of those aggregated aspects are alpha-synuclein. And in fact, Lewy bodies and Lewy neurites are what we call pathological hallmarks of Parkinson's disease, meaning that if we look in the brain of people with Parkinson's disease, and actually the way that we have defined Parkinson's disease is that if a brain is looked at autopsy, if somebody truly had Parkinson's disease, they will have Lewy bodies and Lewy neurites in their brain. And these are real brain pictures as opposed to the cartoon, just to show you what they look like. So this here, where the arrow's pointing, is a Lewy body. What it's sitting in is actually a cell that makes dopamine. Same with down here. This is a cell that makes dopamine, a brain cell that makes dopamine. And this pink blob here, kind of similar to this pink blob, is a Lewy body. This here is also a Lewy body, but not in a cell that makes dopamine, actually a cell that sits out in what we call the cortex, or the higher areas of the brain. And here, this squiggly brown line represents what's shown here, that is a Lewy neurite. So it's actually aggregated alpha-synuclein sitting in some of these long processes of the brain cell. Why is it problematic if alpha-synuclein, why is it that we call these pathological forms? Is that there's a lot of, this is getting back to the laboratory evidence, that there's a lot of laboratory evidence that tells us that when these events happen, it's very detrimental to a brain cell. So here's another picture, another cartoon. This is a different brain cell. And what it's showing is a variety of different aspects of the cell that are required for the cell to stay healthy and stay alive. So there's mitochondria that give it energy. There's all of these systems, the proteasomal system, the autophagy system, that actually take care of, they're basically waste disposals in the cell. So if there's bad proteins or bad fats, it will actually dispose of them. And we know that alpha-synuclein in some or all of these different pathological forms can disrupt many of these different aspects. And so it can cause a dysfunction to the mitochondria, so the cell doesn't have enough energy. It can affect the way that cells handle protein stress. It can actually result in these garbage disposals not being able to dispose of the garbage. And we know also that it can enhance inflammation in the brain. It can affect other proteins to start aggregating, and in some cases, people with Parkinson's disease can actually have proteins that we think of that are associated with Alzheimer's disease, such as amyloid beta, actually aggregate, or a protein called tau actually aggregate. And then it probably affects the communication between cells as well, because brain cells in particular, need to communicate by sending out chemicals to each other. And like I mentioned, alpha-synuclein is involved in the release of brain chemicals. And so when that system gets affected, then brain cells also have difficulties communicating. So as a consequence, alpha-synuclein has been a very, very active area of investigation to try and find a disease-modifying therapy, because if you could in some way reverse all the detrimental effects it's having in a brain cell, then you could imagine you could actually save a cell, or at the very least make it die at a slower rate. And knowing what we know about alpha-synuclein, some of the strategies that are thought to be able to have a positive effect are shown here in this cartoon. So this is another brain cell, and this is a brain cell that it talks to. And so one could prevent alpha-synuclein from being actually made in the cell. And then that way, if you have less alpha-synuclein, you're going to have less problematic alpha-synuclein. You could imagine having ways to actually prevent aggregation, or actually break down the aggregates. And what we also know about alpha-synuclein, what's thought, is that a bad alpha-synuclein or a harmful alpha-synuclein in one brain cell can actually move to a neighboring brain cell and turn the alpha-synuclein in that brain cell into a harmful form. And so if you can actually stop this propagation or harmful synuclein from one cell moving to a healthy cell, then you could also see how you could potentially stop progression. And so just as some examples, these are 3 different drugs that are and have been or are currently in investigation in clinical trials. The green indicates drugs that are still being very actively investigated. And so prasinezumab is an antibody that recognizes alpha-synuclein. And if anybody's been familiar with any of the infusion or immunotherapies, that's what this kind of therapy is. And the expectation is the antibody can actually bind to alpha-synuclein when it comes out of a cell and scoop it up so that it doesn't go into the next cell. Butanatab is a drug that actually can prevent the making of the alpha-synuclein protein. And so this is one other area of investigation. And then minzazolmin is a drug that was developed to actually prevent harmful aggregates of alpha-synuclein to form. And unfortunately, although we're still waiting for the final publication, the company that was overseeing this drug program has found that it hasn't been beneficial in their phase 2 trial. And so we're waiting to see, but the expectation is we'll probably not move on to a phase 3 trial. So that's for alpha-synuclein. I now want to move on to GBA1 or glucose ribosylase or LRRK2, and LRRK2. But as a segue into that, I think it's important for us to just review a little bit around the genetics of Parkinson's disease, because the reason that we're even studying these two proteins, it has come from evidence that has come from genetics. What we've actually learned when investigators have looked into the genes associated with Parkinson's disease. And with that, I thought it would also be a useful opportunity to talk about a Parkinson's Foundation initiative, which is PD generation or PD gene, which is a research project that allows us to learn more about the genetics of Parkinson's disease, and similarly allows us to offer genetic testing to more people, at least in Canada, than we typically would be able to without this research program. And so what PD generation does is it allows for the testing of people who have Parkinson's disease to be investigated for these seven genes that are quite well studied and known to be associated with Parkinson's disease. And you can see here at the top, GBA or GBA1 is the most common PD-related gene, and LRRK2 is the second most common. And as a consequence, not just because they're the most, not just because they're genetic forms, but because they're the most common forms, they are very active areas of investigation. This is also from the PD generation. I think it's useful information for us to see just how common they are. So in the understanding that this is in the context of a research study, that at least what's on the website from Parkinson's Foundation as of this morning, when I added in this picture to this slide, you can see that over 17,000 people have had genetic testing through PD gene or PD generation. Majority of them were negative. As we know, most people do not have a single gene cause for their Parkinson's disease. And we often quote, 10 to 15%. And in fact, PD generation study has proved this to be true, where you can see 12% of people did have an abnormal gene. And again, as expected, but it's nice to see that we actually have numbers to put against this. GBA1 is the most common, and LRRK2 is the second most common. So what is GBA1? So GBA1 is the name of the gene. And what it encodes for, what it actually eventually leads to the making of, is a protein called glucose ribosylase, or I'll call it GCase. And GCase is a protein similar to alpha-synuclein. We all have it. We all need it. It does something in our brain. On the left side of this cartoon is a picture of what a normal GCase does. And you can see some of the similar structures that you saw in my earlier picture. So this is the Golgi apparatus that takes care of proteins in the cell. This is a lysosome, which gets rid of proteins in the cell. And, this represents a regular glucose ribosylase that actually breaks up protein, and in fact breaks up alpha-synuclein, which we need to happen if alpha-synuclein is problematic in our cells. If there's a mutation, so somebody who has a GBA1 mutation, they have an abnormally working glucose ribosylase. And as a consequence, depicted here as a red glucose ribosylase, it gets in the way of a lot of things. So again, it makes it hard for things like the proteasome to get rid of proteins such as alpha-synuclein. It affects the lysosome, which also tries to get rid of alpha-synuclein. It affects the mitochondria. So again, some of the same players that I showed you earlier, because these are really fundamental things for cells to be able to live. And so understandably, some of the approaches would be, well, if having Parkinson's disease due to an abnormal glucose reversidase, which is shown here on the right, is the problem, can't we just give people back a working glucose reversidase? And so one approach that's being investigated is exactly that, basically replacing glucose reversidase. I'll talk a little bit about it at the end, but being able to actually take glucose reversidase and deliver it to the brain using a technique called MRI-guided focused ultrasound, or actually giving a gene therapy that allows the brain to make its own new glucose reversidase by injecting a virus that has the gene therapy into the spinal fluid. Or an alternative approach is just modulating, helping the abnormal glucose reversidase just function better. And so there's an ongoing trial, if not yet started, just about to start, a phase 3 trial for Ambroxol, which is thought to help fold and support the function of an abnormal glucose reversidase. And also this compound here, which is also thought to modulate and enhance the function of glucose reversidase, and so make the abnormal glucose reversidase just work better. Switching now to LRRK2. LRRK2 is a different kind of protein in terms of the way that it's affected in Parkinson's disease. So here in glucose reversidase, like I mentioned, glucose reversidase doesn't work well enough. So it has some inactivity. Whereas LRRK2, this is a protein that has a lot of functions. This is a very simplified version of what it does. But one thing that it does do is it usually has limited activity in the cell. And when it gets hyperactive as a consequence of a mutation in it, then it starts causing problems with a variety of different types of pathways. So this is a Rab protein. It affects these Rab proteins that are involved in many, many intracellular processes in terms of shuttling things properly within a cell. It affects the lysosome again that you've heard about already. And I think what's interesting and areas that are important for us to investigate are that we know that mutations in the gene, so a LRRK2 mutation causes hyperactivity of LRRK2 and problems in the cell that leads to cell death. But interestingly, there's also a large amount of experimental laboratory evidence that also says that even if you don't have a LRRK2 mutation, if you've been exposed to certain toxins in the environment, such as pesticides, that itself can actually lead to hyperactivity of LRRK2, and then the consequences of that being neurodegeneration. So areas of investigation for disease-modifying therapy to target LRRK2 includes inhibiting it. Like I said, it's overactive, so there is an ongoing clinical trial of a drug to just try and dampen down its activity. And then there's also approaches to actually not necessarily change the activity of LRRK2, but just make it so that the cells make less of it. And that's with techniques called ASOs, or antisense oligonucleotides, similar to therapies that are being developed or started to be developed in Huntington's disease and ALS, where an ASO is injected again into the spinal fluid, and that prevents the cell from making LRRK2. So for the last 5 minutes, I just want to now touch on some of the research challenges and how this impacts our future directions. And I thought it would be useful to focus on areas of brain delivery and biomarkers. And brain delivery, I think, is particularly important. As I mentioned at the beginning, sometimes people will raise up their hands and say, oh, it's too hard to find treatments for the brain because it's so complicated. And often with that is the idea that it's a challenge because since the brain is in a privileged space, because we have a blood-brain barrier that prevents things from actually getting into the brain and harming the brain, it also works to make it harder to get things that we want to get into the brain, like drugs, to help the brain, a challenge. And so even if we can find things that work really well in the laboratory, in, for example, cells that we've grown in a dish, um, that kind of environment doesn't take into account the protection of the blood-brain barrier. And so that always has to be considered when we're thinking that we want to try and get this treatment to actually be able to help humans. And then biomarkers I'm going to touch on as well, because there has been a lot of talk about biomarkers over the past couple of years. And I thought that there may be some questions around it, so I at least wanted to have a little bit of a segue into those questions. So starting with the brain delivery aspects, I already alluded to the blood-brain barrier, and this is a bit of a cartoon of it. So, if we have the brain here, within the brain, there are blood vessels that course throughout the brain, and it's through blood vessels that things are delivered. And so normally things can actually easily cross if necessary. But in the brain, there's what's called the neurovascular unit, where if this is the blood vessel, and this is where blood flows through this lumen of the blood vessel, there's a substantial number of almost walls for things to pass through to get into out here, which is the brain. So there's the wall of the blood vessel, of course. There are these other cells called pericytes, which wrap around the blood vessel to try and prevent things from leaving. And then there's things called astrocytes that actually have what we call foot processes that also wrap around the blood cell and try and prevent things from leaking out of the blood cells into the brain. So that's the blood-brain barrier. And typically, small things, so small molecules, small molecules that are drugs can sometimes get across the brain, but bigger things like viruses or stem cells or even more complicated drugs don't always get their way across the brain. So, for example, dopamine can't cross the blood-brain barrier. It only can The only way that we can get dopamine into the brain is to have levodopa, which crosses through the blood-brain barrier, be able to be turned into dopamine in the brain. So there are a number of ways that we can get things into the brain. I'm just going to put up two examples here because they relate to two of the potential disease-modifying therapies that I mentioned earlier. So one form of delivery is what we call intrathecal delivery. And so thecal is basically the sac that surrounds the brain and the spinal cord and holds in the brain and the spinal cord and the spinal fluid. And so if we can breach that sac and actually put things into the spinal fluid or the cerebrospinal fluid, then the cerebrospinal fluid, which bathes the brain, can allow us to get things to the brain. So glucose riboside replacement, I mentioned, is one strategy to try and help when glucose riboside is working abnormally. And so a viral delivery of a gene therapy is being investigated. And viruses don't cross the brain, and we don't want them to cross the brain because that's how we don't get viral infections into the brain. But the way that it's being delivered for this therapeutic is basically by injecting it into the cerebrospinal fluid here high up at the base of the brain. And then the alternative type of approach, which is LRRK2 therapy to bring down its levels and make the brain cells make it less, is using something, as I alluded to earlier, called an ASO or an antisense oligonucleotide, which again will not cross the blood-brain barrier, and therefore it's being delivered in the same way as if anybody's had a spinal tap, entering the thecal sac that way and then injecting in the ASOs there. Another way to get things into the brain is a little bit more sophisticated, and that's using a somewhat an old technology that has that's basically been finessed with some new technology. So ultrasound is an old technology. I'm sure many of you have had an ultrasound before, but new hardware and capability and software capabilities have allowed the use of ultrasound to be focused in a very, very specific area and with the use of an MRI to see where the ultrasound beam is being delivered. And there's a variety of ways that it's being used, but in terms of drug delivery, one way that it's being used is to actually help open up the blood-brain barrier temporarily. And the way that this works is that little microbubbles are given into the bloodstream. And when these microbubbles shake around, they can actually break up that kind of wall that I was showing to you that makes up the blood-brain barrier temporarily to allow things to move from inside the blood vessel out into the brain. And what shakes it up is a little bit of ultrasound. So we have been studying whether or not we could use this technique to actually deliver glucose cerebrosidase, which is a big protein that you can give to people intravenously, but it doesn't get into the brain. And so we've been trying to couple that together with this focused ultrasound in this MRI machine to see if we can get that into the brain. So for the last minute or so, I want to touch on biomarkers, and I want to bring you back to this slide here of the symptoms of Parkinson's disease and then this underlying pathobiology. And currently, right now, from the point of view of clinical trials, from even making a diagnosis, we very much rely on symptoms. What do we see? What can I measure? What can I observe when I get people to do tasks? What scale scores do they have? That sits up all here in the symptoms. And what we've been lacking for a very long time is actually biomarkers which tell us something about the underlying pathobiology. So what's happening underneath? And again, if I go back to this earlier slide around multiple sclerosis and the successes that multiple sclerosis has had, inspired by this, a couple of years ago, I brought together colleagues in the multiple sclerosis field together with my colleagues in the Parkinson's field to have a meeting to discuss what what have allowed for the successes of multiple sclerosis and what can we learn from each other. This is not a conclusion from the meeting, but came from the meeting. Just I had to sit down and look through the histories of both of the diseases. So this is the timeline of multiple sclerosis over the centuries, basically, and this is Parkinson's disease. And in many ways, they parallel each other. They were described at a similar point in time. Each had to develop some laboratory models. Each had to develop different scales. But one thing from our discussions that came about was really the importance of this use of MRI in multiple sclerosis as a biomarker. And the advantage in multiple sclerosis is that when people have a relapse or an attack, that this is often reflected by inflammation that can be seen on a relatively routine MRI brain. And so for multiple sclerosis, having a biomarker of inflammation, which is their MRI brain, was really helpful for them to then start to study anti-inflammatory drugs, which is what most of their therapies are. And in Parkinson's disease, we're starting to get there, starting to have actual biomarkers of underlying pathobiology. And again, going back to the examples that I showed you earlier, if I just focused on alpha-synuclein, GBA1 or GCase and LRRK2, we now have a biomarker of alpha-synuclein aggregation. So we can test people and get evidence that not by doing an autopsy and looking at their brain under the microscope, like I showed you those slides of, but actually test them to see if there's alpha-synuclein aggregation. And so then that allows us now to say, okay, well, if somebody has alpha-synuclein aggregation, we should be testing alpha-synuclein therapies in them. And I think close at its heels are similar biomarkers that will help us to look at glucose cerebrosidase. So, I mean, now we can look at gene abnormalities, and that in a way is a biomarker, so that we can test people who have a GBA1 gene abnormality. We can test these GBA1 or GCase therapies, and similarly, people who have a LRRK2 gene abnormality, we can test LRRK2 therapies. But I think that really to expand out these therapies to not just those who have a gene abnormality is having a biomarker so that if somebody doesn't have a gene abnormality, but we can measure that their glucose cerebrosidase is inactive or their LRRK2 is hyperactive, then we know that they could be people who could be in clinical trials and eventually given therapies that affect each of those underlying pathobiological mechanisms. So with that, I just close out just to say that I hope that this has been informative. The purpose, again, was really to help us understand where the current state of disease-modifying therapies are for Parkinson's disease. We reviewed some of the areas of investigation that I think are particularly hot, including alpha-synuclein, GBA1 or GKs, and LRRK2, and that there are challenges, of course. I don't think insurmountable challenges, and two major ones that you may hear a lot about in the near future are how are we going to get promising therapies into the brain and not be restricted by the challenges of the blood-brain barrier, and what biomarkers are we going to have out there so that we can test people, identify what kind of therapy they need, and then enroll them in the right kind of trial, and eventually, when those trials are positive, give them the right kind of therapy. So with that, like I mentioned, I'm talking to you from Toronto, Canada. We have a sunny day like this right now, so I thank you very much for your attention, and I'm really happy to take any questions.
Thank you so much, Dr. Kalia, for offering us an in-depth overview of what disease-modifying therapies means and what we can see some projections in the future for Parkinson's disease. So thank you for outlining that for us. We do have a few questions, some that aren't related to disease-modifying therapy, so I'm just going to offer the ones that are to start. We got a comment from one of our participants sharing that it's very interesting, the comparing and then contrasting with MS, and I'm curious if there are any of the anti-inflammatory drugs for MS that are showing any promise that could be applied to Parkinson's disease treatments.
Yeah, and I think that's a great question. And the meeting that we had, of course, with individuals, experts in multiple sclerosis and Parkinson's disease, I think it was timely, and we probably need another meeting because it's really relatively new. I think that we think about inflammation and Parkinson's disease. If you think 2 decades ago, much of the thinking was neurodegeneration leads to neuroinflammation, and the neuroinflammation is really just a bystander of neurodegeneration. But now we know that neuroinflammation is probably a player here, and if we could affect neuroinflammation, then we might be able to reduce neurodegeneration. And so I think we're very early days. I think looking to what multiple sclerosis has is going to be a really important area of investigation, especially since many of these drugs have had many years of experience. They've been treated on people who have neurological disease. So there's not one in particular, but I do think keep our eyes in that area, because I think we're going to see some advances there. And I think the other important piece around that I learned from that I guess gives us a little bit of pause as well, which I learned from my multiple sclerosis colleagues, is they are quite confident and happy and with their armamentarium of treatments that affect inflammation. And they have many therapies and lots of evidence to show that they can really make a difference on neuroinflammation. But in multiple sclerosis, there's neurodegeneration too, and they're still struggling with being able to have good therapies that actually stop that process. So even when they are able to really reduce relapses due to inflammation, it doesn't necessarily stop the neurodegeneration that happens in multiple sclerosis. And so one wonders if there might be some shared learning between multiple sclerosis and Parkinson's disease around neurodegeneration.
Thank you for that, and also sharing how we can explore the learnings of another disease and apply what the curiosities are from our researchers there, what they're finding there, and how that could really influence the research for Parkinson's disease. A question about finding these, and I'm going to use the words from our participant, these reputable high rigor clinical trials that we as participants can support researchers in further exploring disease modifying therapies. Do you have recommendations on how we could find these types of clinical trials?
Yeah. So I mean, I think that the usual resource. So there's a couple of. So the Michael J. Fox Trial Finder is one resource out there. And I'd say your clinician is another very important resource. I know not everybody has a movement disorders neurologist as their care provider. If you do, they will often have their finger on the pulse of what's going on, and if not, have colleagues that they can closely call up to find that out. And then similarly, I mean, if you don't have a movement disorders neurologist, I think putting it to them in terms of uh, reaching out to their colleagues to find out what they think is rigorous and valuable to participate in. Also appreciating, unfortunately, just the way that practicalities are. You know, I wish in our center we had the availability of offering every trial that I'm excited about to patients here. But unfortunately, not all clinical trials end up at all centers. I can tell you, even from a country's point of view, there are some clinical trials that actually never see a site in Canada, and I wish that for patients. So there's that piece too, that you want to find a good clinical trial to participate in if that's something that you want to do. But even if you find one that you're really excited about, it's also a matter of, can you actually participate in it? Is there one close to you? Does it make sense? Is there even something like that going on in your country? So yeah, I say Michael J. Fox Trial Finder, and then really speaking to people locally as to what they think is valuable, and what you can actually participate in.
And recognizing the barriers that are there, whether they're physical boundaries or location, even your enrollment, like what's your story? There's a lot to consider when looking into clinical trials. So thank you for guiding us towards the trial finder, and also opening the invitation to have the conversation with our providers. Just looking at the time, I've got a couple of questions that aren't necessarily related to disease modifying, but we do still have like 2 or 3 minutes.
Yep.
And I'd love to just have the opportunity while we have you, Dr. Kalia, to ask some of these. So, therapy and medication related. Jennifer has shared that they've read a few articles about NAD and psilocybin as Parkinson's treatments. Can you speak to these two compounds and their applications that might be potential for Parkinson's disease?
Yeah, and so I'm not quite sure what you were reading about. My understanding, so NAD is the hope around NAD is that it's a bit of a energy booster to the cell. And I'm not aware of actually it being currently in a very large clinical phase trial. But at least a couple of years ago, I know that there were smaller trials. And so I think we keep our eye in that I wouldn't say go out and purchase it and take it yourself. I think that for one, you want to see the rigor of it being valuable for you to take, both from a risk point of view and also just your pocketbook point of view too. So I think that is an area that will probably continue to be investigated. And then psilocybin, if I'm correct, is probably being more investigated for mood-related issues related to Parkinson's disease. And again, I'm not aware of any, and that's a little bit more symptomatic therapy. So I'm not up to speed on what clinical trials are being done for that, but I know that it's being investigated in a whole host of different areas. And so again, I think it's an area to keep your eye on, because I do think we'll probably hear more, but nothing again right now that I'd say go out and self-treat.
Thanks for giving us that cautionary reminder when considering clinical trials and observatory trials or anything else that might be a trial in research, whether it's mood-related, symptomatic, or disease changing. So I appreciate that cautionary approach to coming into and making decisions of what we choose to engage in. I'll offer one more question. Is neuroinflammation aggravated by stress? Could you just speak to that really quickly?
And that's a wonderful question. Uh, so stress is is we need to understand more about stress. I think is the bottom line. I have a colleague at my research institute who is really just interested in stress and all, understanding stress, and it's this goes back, I think, again to the cross pollination of getting people from different areas to kind of get into a room together, and And as a consequence of that, I've started to think about stress more and listen more to what my patients say about stress. And I know that probably everybody on the call can tell me stress is a major issue in Parkinson's disease. It makes my symptoms worse. I've had patients who've had a stressful event that is actually what, they didn't have symptoms of Parkinson's disease. They had a very stressful event, and that it seemed to be the trigger for what led to their Parkinson's. And so I think it's a great question. Does stress lead to more neuroinflammation? My hypothesis would be, yeah, probably. It probably leads to a whole bunch of things. And I do think that this is an area that we really have to investigate more. Of course, often, like I say with patients, life is life, and if we could all live a stress-free life, wouldn't that be wonderful? Unfortunately, we can't. So finding ways to reduce stress that doesn't need to be in our life, finding ways to stress manage. But I think we also need to really understand the biology of stress and how it interplays with those pathobiological mechanisms in Parkinson's disease. I'm willing to bet that it aggravates them.
And understanding what stress is, right? So are
Yeah.
we talking physical stress, mental stress, energetic stress? There's so many components behind stress that we still are learning as a human population across the globe. So thank you for offering a little bit of insight on how stress impacts us, just not only as humans, but humans living with Parkinson's disease. Dr. Kalia, this doesn't happen often, so I'd love to read these words. Someone wrote into the Q&A. This has been a great presentation. There's been a lot of information, and it was presented really well, and that they learned a lot. So thank you so much for sharing your expertise, the years and time and compassion that you've put into understanding Parkinson's disease and helping us move treatments forward does not go unrecognized. So thank you for all that you do.
Thank you very much. And thank you for spending the afternoon with me.
And thank you everyone for joining us today and for being curious about disease modifying therapies. I want to celebrate Dr. Kalia's presentation in the way that she broke it down for us to understand what it means to be a disease modifying therapies and what we can look forward to potential treatments in Parkinson's disease. And thanks again to all of you for your curiosity. Before you go, I'd like to share some resources and opportunities with you. A reminder about our other PD Health at Home virtual programs. Each week, we offer sessions tailored to support people with Parkinson's and their care partners, including Mindfulness Mondays, which is a great resource for the stress that we're just ending on, speaking to. Our Wellness Wednesdays, which you've found us, Fitness Fridays, and expert briefings. We also have a Spanish language series, Y Salud en Casa. Be sure to visit our website to see what's coming up next and explore archive videos on YouTube. Learn more about those upcoming virtual programs, and you can register at parkinson.org/PD Health. And before we wrap up, we just have one last favor to ask you. Your feedback is essential to shaping the Foundation's programming. The reason why Dr. Kalia's presentation was here today is because the Foundation received multiple requests to learn more about disease-modifying therapies through your voice. So please take a moment to share your thoughts and let us know what topics you'd like to see in the future. As the webinar concludes, a survey will pop up on the screen. It takes less than 3 minutes to offer us what you'd like to see more of in future webinars. And if we didn't get to your question or if new ones come up as you process the information, know that we're here to help. You can visit our website at parkinson.org for a wealth of resources on Parkinson's disease or reach out to our expert team directly. You can call our helpline at 1-800-4PD-INFO or email us at helpline@parkinson.org. Thanks so much for joining us today. Together, we can navigate this path and continue building a brighter future in community, in research, and in movement. Take care and be well. I'll see you soon.